Hi, I'm Srishti Nayak. I'm a postdoc research fellow at Vanderbilt University Medical Center in the US. And today I'm presenting work that was done as part of my dissertation research at Boston University with Amanda Tarullo. My talk is about how home language mixing exposure in children is associated with their neural markers of attention or executive function. Central to my work is the idea that linguistic and cognitive outcomes of bilingualism are fundamentally intertwined, or as this quote says, intimately interconnected and mutually interdependent. In fact, we now have a lot of evidence that when bilinguals control or manage their languages, they're also engaging brain areas underlying executive processing or other cognitive processes. Just a refresher, I'm going to be using the term executive processes, and by this I mean the constellation of cognitive processes that allow us to plan, organize, think, regulate, and so on. Not only are bilinguals engaging their executive control brain areas when managing their languages, the extent of bilingual language control abilities is associated behaviorally with executive control abilities as well. And this is true during comprehension, during production, and it's true in both adults and children. Of course, we know that when we talk about bilingualism or the experience of being bilingual, we're actually talking about a bunch of different cognitive experiences and linguistic experiences. And these vary along a spectrum depending on your own environment. So for example, how many times are you actually switching from one language to another language, from your first language to your second language and back on a typical day or in a typical week? Or how often are you actually mixing up your two languages or code switching between them at the level of words or sentences or even conversations? So this idea that bilingual experiences are varied and also that the variability in bilingual experiences may lead to different kinds of neural adaptations is now a pretty robust idea with lots of evidence behind it, to the point where we now have 5 separate sort of neural models of how specific patterns of language exposure, language learning, language use, or even interactional context might lead to specific adaptations in the brain. A lot of that data from the neural adaptations models that I just showed you comes from adults though. So as a developmentalist, I'm really interested in thinking about children and particularly the preschool to school age transition, which coincides with the most rapid development that we see in executive function processing across the lifespan. So what about how bilingual experiences shape executive processing in children? So the research question I'm talking about today is, are individual differences in bilingual children's language experiences associated with variability and individual differences in their executive processes, by which I mean neural processing? Specifically, when I say language experiences, in today's study, I'm talking about home language mixing experiences. And when I say executive processing, I'm talking about neural mechanisms related to specific kinds of executive processes. And the measure that I looked at are a specific event-related potential called the centroparietal B3 waveform. So why language mixing specifically? Language mixing is very common in bilingual families. I'm sure anyone whose bilingual family speaks multiple of the same languages as them can attest to this. This phenomenon of code switching or translanguaging or language mixing, like other bilingual language habits, it's really hard to quantify and measure. But for this one, we actually have a validated measure that exists already, which we'll talk about in a second. And really promisingly, in infants, home language mixing is associated with attentional control, attentional monitoring, and inhibitory control, which are all executive processes that we're interested in. But again, this promising data comes from infants, and I am interested in looking at that preschool and school age time. And just to say a little bit more about how we're measuring executive processing here. So we're using the centroparietal P3 event-related potentials, which are sometimes known as P3Bs. And one of the reasons is that this event-related potential, or ERP, has already been associated with executive processes in children of a similar age than who we were testing. And namely, P3s have been associated with inhibitory control, context updating, task switching, and context monitoring demands, which are all sort of within that realm of executive processes that I'm interested in. And just a quick refresher, in case anybody needs it, event-related potentials, the special thing about them is that you're measuring the brain signal time-locked to when the stimulus is presented. And you get millisecond precision in terms of when things are happening in the brain. And so in order to get a P3, if you look at this plot over here, this is just a schematic, it's not real data, but just to give you a sense. So a P3 essentially refers to the third positive deflection after the stimulus presentation. And it's sometimes customary for ERP schematics to have positive below the line. So this is the first positive, second positive, and third positive wave. And we then sort of average across many trials per person and average across participants in order to get the sort of averaged signal. So for our study, we brought children into the lab and we had them play a computer game called the Animal Size Troop, an adapted version from Bryce et al. 2011. And we collected EEG data while they did that. And so to give you a sense of our participants, on the right, we had 33 bilingual kids who also had usable EEG and all the questionnaires that we needed. So we actually had more, but you do get data attrition in EEG studies with children. They were all 6 to 8 year olds, and they were all dominant in English as reported by their parent. But their second languages were heterogeneous. So we weren't looking at a specific L2 group. Rather, there were many languages as represented in the greater Boston area. And then taking you back to the game for a second. So the game is basically which animal is bigger in real life, and you have a congruent condition and an incongruent condition. So in the congruent condition, the animal that's bigger on the screen is bigger in real life. So you select the button that's on the same side of the screen as the animal that's bigger in real life. And in the incongruent condition, the smaller animal on screen is bigger in real life. So you find the button and you click it. And then while they played that game, we measured their reaction times on the response pads, their accuracy on 128 trials of this game, 4 blocks with some breaks in between. And we measured their EEGs, and then we extracted their centroparietal P3s, as I mentioned earlier. And while the kids did that, their parents filled out the home language mixing questionnaire. So the researcher pre-fills the questionnaire with the home language. I often start a sentence in English and then switch to speaking, for example, Hindi. And the parent chooses from 1 to 7. And it's a very straightforward and simple parent questionnaire. The only thing is that we're asking about language behaviors in child-directed speech specifically. And the family ends up with a score from 0 to 35. So to take you through the results, we first did a sanity check that our game, the Animal Size Troop, does in fact elicit P3Bs in our age group. So we had a larger sample of 84 kids that was both monolinguals and bilinguals, and this is the grand average of the data from all of those 84 kids. So as you can see in the shaded gray area, the task does elicit the expected P3Bs, and as expected, it elicits a stronger P3B response, i.e. more positive peaks, in the incongruent condition, which is the Stroop condition, where you have to sort of correct for the fact that the smaller animal on screen is the bigger animal in real life. So once we had that sanity check out of the way, I looked at the relationship between home language mixing scores and P3B amplitudes and latencies. And the results were both surprising and really exciting and promising. So firstly, we only found a relationship with home language mixing scores on the congruent condition. Secondly, we did find a negative relationship between P3B amplitudes and home language mixing. So children with the most home language mixing, as reported by parents, had the lowest P3B amplitudes. Now, there are many ways to interpret that, but before we get into interpretations, I want to say that, this thing kind of stood out to me here, where even though it was a linear relationship, I wanted to look into the fit a little bit more. So I tested both linear and quadratic, which isn't shown here, and cubic relationships, and I found that the cubic relationship actually had the best R square, and quite a significant jump from the linear one, and that sort of makes sense if you look at the trend. Now, I do acknowledge that this is only 33 children, so there may be some overfitting here, and I only looked at the sort of default assumptions of linear, quadratic, and cubic. However, I do want to think about what this kind of relationship could possibly mean zooming out theoretically. So one idea that I've been thinking about is related to a neural practice effect at the lowest ends of language mixing, a neural effort effect in the sort of middle range, and a neural habituation effect on the high mixing kind of end. And this is speculation, but just thinking about the fact that the practice, the effort, and the habituation happens kind of at the level of the language mixing, and then what we are theorizing is that it spills over, in a sense, to your neural mechanisms of shifting and switching and keeping up with dynamic input in other domains other than language. So it's just an idea, but I'd love to hear what other people think about it. I'm still trying to develop the idea fully. So some future directions I'd like to keep thinking about. First, we found that the language mixing questionnaire can actually easily be used in parents of older children, but technically it was designed and validated for parents of infants. So it would be nice to have some formal evaluations for families with school-age children. Secondly, while we found a link between home language mixing and P3Bs, which I'm really excited about, we certainly need replications and larger samples. And even though 33 is a decent sample for EEG studies with children, for individual differences approaches, you really need larger samples to really capture variability in a meaningful way. And last, I'm pretty curious about why some neural markers of executive function, but not others, might be associated with something like home language mixing. For example, I tested the N2s that were elicited from the same study and the same game, and those were not at all associated with home language mixing. So just a thought. And just a reminder that our broader goals are to study global and diverse, rich patterns of home language experiences in different multilingual contexts. So we really want to always think about how we can do this kind of work in different contexts. And ultimately studying how language experiences and individual differences in language experiences support neurocognition and neurocognitive development. Even studying this in bilinguals can actually help us understand this relationship more broadly between language and cognition. And with that, I'd like to thank the team at the Boston University Brain and Early Experiences Lab, our funders, this conference, and all the families that make our studies possible. Thank you, and I look forward to your questions.